Hello class, my name is Elias Bergel. This is my visual presentation on chapter 38, section 6, the diverse visual receptors of animals depend on light absorbing pigments. It all starts with photoreceptors, okay? There's a vast and varied amount of light detectors across the animal kingdom in invertebrates and vertebrates. And all these light detectors have some sort of photoreceptor. And what these do is they detect the radiation that we see as visual light. There's common and shared genes between a lot of species. You wouldn't think share genes, and this is kind of pointing at evolution and a common ancestor amongst a lot of animals. There are many types of eyes and light detecting organs. There's the ocelli, the compound eye, and the single lens eye. That one's real important. The ocelli, or the ocellus, or ocelluses, that's plural. If I'm wrong, sue me. It is a basic light detecting organ, and if you have two, the differential between the action potentials help guide an organism to shade. As a pale person, I really appreciate this trait. Sunblock is not always around. The compound eye is insects. It is in insects, crustaceans, and some polychaete worms. It's multiple light detectors. Think of a fly, how they have just a ton of little eyes, and this helps them make out basic sight and movement. Like my roommates moving over and over through this space, even though I said I needed it. This is my 8th take. Single lens eye. This is in some invertebrates as well as vertebrates. Invertebrates like an octopus. Vertebrates like my buddy Colt holding the camera with his baby blues. It's very complex. Before I move on to the certain aspects of the single lens eye, there are some evolution deniers that use the complexity of eyes as a point to say that evolution just couldn't feasibly guide us to this point. But there's shared genes. According to National Geographic, the work of Hiroshi Suga strongly suggests all animal eyes share a common origin, whether they belong to a human, insect, octopus, or jellyfish. I didn't even know jellyfish had eyes. Okay, and if you break it down, it's not that crazy to see the evolution. You get an ocelli, or a single photoreceptor, through evolution, and it can detect light. Okay, I don't know if you can see the flashlight real well, but it detects light. Then you get like a plane of it. Okay, and that starts to curve, then you can detect light in directions. Okay, and maybe that gets a little better, gets a little more round, like a pot. And then if you poke a hole through that, it's basically a single lens eye. You know, it's not that crazy to think of, going from just the most basic unit to what we have now. Key parts of the human eye. There's the sclera / cornea. The sclera is the outer membrane that's transparent. The sclera is the outer membrane. It makes the transparent cornea of the eye. And then you have the iris, which is the colored part of the eye. This opens and closes to allow more or less light in. You know how your pupils dilate, and that is formed by the choroid. The pupil, as we just mentioned, is that space that the light enters, okay? And in the back of your eye, past all the fluid, is the retina. And this is where all your photoreceptors are. And once light hits that, that's what allows us to see, because that's what all photoreceptors are. And the highest concentration of those photoreceptors is our fovea. And you can see that here, or on the PowerPoint, if you follow along. And that creates the center of our visual field. In contrast, the optic disc makes a blind spot in our vision. And the optic disc is where the optic nerve leaves the retina, okay? Because once the retina receives the information, it sends it through the optic disc to the brain. Woo. Rods and cones, those are our photoreceptors. They are, the rods are more light sensitive, and they really help us see in the dark and help us distinguish shape. They're on the retina, of course. And cones are a little less light, well, they're a lot less light sensitive, but they help us see colors, okay? They help us see visual pigments called photospin, and we see three. We see red, we see green, and we see blue. Red and green are both linked to the X chromosome, so red and green color blindness is quite, is typically linked to men much more, because we only have one X chromosome. Women have two. And even in blue color blindness, which isn't linked to the X chromosome, men tend to have it more. Just going back to the evolution aspect, rats experience color blindness too. They see in different shades than us, but according to the study done for the American Journal of Physiology, a rat does not discriminate under certain conditions, and his vision is closely analogous to that of a total colorblind man. So they see a lot like we do. Transduction, this is kind of complex. In the dark, we have a lower membrane potential. In order to see, it's negative 40 millivolts. And this is because we have constant flow of sodium ions, positively charged. And this allows glutamate to be produced so we can see in the dark. In the light, we don't have that glutamate produced. That's because rhodopsin activates once light hits it, and then it combines with, it activates transducin, and that combines with phosphodiesterase, which causes the sodium ions to stop flowing, and then that glutamate to stop being produced, and our action potential goes down to negative 70 millivolts. When we see, it flips from right eye to left side of the brain, from the left eye to the right side of the brain. And we actually have dominant eyes, like we have dominant hands, or we are right brain or left brain thinkers. According to a study done for the Journal of Motor Behavior, when information from the dominant eye was available, the test subjects were led to have enhanced accuracy and precision in comparison to when they had input from the non-dominant eye available. Other than that, we see nearly 180 degrees, not quite. And what helps us in our periphery are those cones that help us make out shapes. So not quite 180 degrees. And these are my references. Thank you.